Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. So in last class, we learned about actin filaments, which are also known as microfilaments. So we were learning about different cytoskeletal filaments. So we have already covered one of them, and now we are going to learn about intermediate filaments. So as learned in the introductory class, intermediate filaments, they are made up of intermediate proteins, and this kind of filaments, they are less dynamic, they are less flexible, and they are rod like. So unlike microtubules, which have hollow structure, which are tube like, intermediate filaments are rod like or rope like, and that is why they provide the mechanical support to the cell. So they have 10 nanometer diameter, which is moderate, which is intermediate in size. Like the actin filaments, they are 7 to 8 nanometer in diameter, while microtubules are of 25 nanometer in diameter. So the size of intermediate filaments lies in between. That is why that is also a reason to call them as intermediate filaments. So they are made up of different types of intermediate proteins. So there are 6 major protein types. We'll learn about them as well. Before that, let's learn about the basic structure of intermediate proteins. So intermediate proteins have three domains: the N-terminal domain, the C-terminal domain, and the alpha helical domain. Okay. In all of those 6 types of proteins, this alpha helical domain is conserved. So this is the conserved domain, while the N-terminal and C-terminal region differs, and that is why we have 6 different types of intermediate proteins. So this polypeptide, this N-terminal to C-terminal polypeptide, it forms dimer in parallel orientation. Means the N-terminal of one polypeptide will, it will pair up with the N terminal of other polypeptide, while the C terminal of this one polypeptide will it will interact with the C terminal of another polypeptide. So this is what we call as the parallel orientation. So two intermediate proteins, they come together, they form a dimer, and then though two dimers, they come together in anti-parallel orientation to form a tetramer. So this is a very important detail, that dimer is formed in parallel orientation, but the tetramer is formed in the anti-parallel orientation. Then the 8 of such tetramers, 8 units, 8 tetramers, they come together in parallel fashion. So normally, so this is one tetramer, this is two tetramer, okay, this is the third tetramer, fourth, fifth, sixth, seventh, and eighth. So 8 tetramers come together to form a protofilament. Okay, and 8 such protofilament, they come together. So these are the 8 protofilament laid together, and they roll in a certain direction, okay, in a certain direction to give one intermediate filament. Okay, so if you try to view this from above, so this is top view. This is how the 8 protofilaments will look like when they come together to form an intermediate filament. And this is how there's no gap left, and that is why it is a rope-like structure. Multiple threads are coming together to form a rope, and that is why intermediate filament is less dynamic. It is a bit rigid, and it is going to provide mechanical support. So as discussed earlier, there are different type of intermediate filament proteins. So I've listed them down here. The very important one and the first of them are keratins. So there are some acidic keratins as well as neutral or basic keratins. They are found in the epithelial cells. So they give the robustness to the epithelial cells, the keratins. So this is an important intermediate protein. Then there is vimetin. This is found in fibroblast, which secrete fibers, as well as in WBCs and some other type of cells. So when we were discussing about cancer in cancer biology, when we discussed about epithelial and mesenchymal transition, I hope you remember that vimetin was a very important factor in the epithelial to mesenchymal transition, like the E-cadherin to N-cadherin transition, as well as vimetin transition is also seen in EMT. Okay, I hope you remember that. If you don't, please visit that lecture. Then there's other protein known as desmin. It is found in muscle cells. Then there are some glial fibrillary acidic proteins. They are obviously found in glial cells. So all of those are the intermediate filament proteins. And then There are certain intermediate filament proteins that are found in neurons, such as NFL, NFM, NFH. So these are also, collectively, they are also referred as neurofilament proteins. Then there are nuclear lamins. So as discussed in the introductory class, the major function of intermediate filament is to provide mechanical support. So this mechanical support is also provided not to just cell but as well as to the nucleus. So inside the nucleus, there's a mesh formed by the intermediate filament proteins, and that is what we refer as the nuclear lamina. This nuclear lamina is found in almost all of the cells, and this nuclear lamina is made up of the proteins which are referred as the nuclear lamins. Okay, so this is a very important point to note down. Then there are proteins such as nestins. Those intermediate filament proteins, they are found in stem cells. So specifically the stem cells of the central nervous system. So with this, I conclude the lecture about intermediate filaments. In next lecture, we will learn about microtubules. Till then, see you. Bye bye.